The true false, I'll only cover the false. Question 2. The production of ATP by substrate level phosphorylation occurs during cellular respiration. That's false because that is oxidative phosphorylation. 4 is false. The outer membrane structure of gram-positive bacteria consists of lipose polysaccharides. Gram-positive bacteria don't have an outer membrane. Number 9 is false. Microwaves are an effective way to kill bacteria. They are not. 11 is false. Gram-positive cells have a very thick layer of peptidoglycan surrounded by an outer membrane. Again, they don't have an outer membrane. 12 is false. Cells located in the center of a 48-hour colony are growing at an exponential rate. No, they're probably starving. 16 is false. Endotoxins are an integral part of the outer membrane of gram-positive bacteria. Again, the theme here, gram-positive bacteria don't have an outer membrane. 18 is false. The transfer of fertility plus plasmids requires contact with another fertility plus bacterium. No, actually contact with a fertility minus plasmid. Okay, 21. Bacteria do not use this process as a means of transport across the cell membrane. Endocytosis. 22. This process occurs in the interior lumen of the mitochondria. That's going to be D, the tricarboxylic acid cycle. 23. In the absence of oxygen, this pathway is shut down in aerobes. That's going to be C, respiration. 24. Which of these methods cannot be used on media? That's going to be D, the oven. 25. Which of the following terms implies ATP production by oxidative phosphorylation? Actually, that's going to be D, all of the above. 26. This type of mutation causes a frame shift mutation. That's going to be B, addition of a nucleotide. 27. The proton motive force produces ATP using B, oxidative phosphorylation. 27. Well, that was 27. 28. Which of the following is a uniquely bacterial molecule? In this case, both of them are. 29. Which of the following is true of the fermentation process? In this case, all of those things are true. Excuse me, none of them are true. I'm sorry. None of them are true. It's D. It is not a transition step. It does not produce energy, and it does not generate reducing power. 30. Which of the following best describes the bacterial genome? A, circular and haploid. 31. DNA can be transferred from one bacterium to another when viruses serve as the vector. That is transduction. 32. A cleaner with a D value of 2 minutes is used on a population of 1,000 cells. How long will it take it to get to one cell? 6 minutes. That's not a choice, so your answer is D. 33. Which of these would have their growth severely inhibited by the presence of oxygen? That would be B, an obligate anaerobe. 34. Which of these methods would I use to sterilize laboratory glassware? Both methods are acceptable, so C.
I have a question.
Sure.
Didn't you say it's oven at 20 Celsius only for the glassware?
Yeah. Not only. See, not only. You can sterilize your glassware in an autoclave as well, and I did make that point. Sorry. 35. Which of the following statements is true of cellular respiration? That would be D, all of the above. Which of the following, 36, which of the following is not true of enzymes? And that would be D also, none of the above. All of those apply. 37, differential stains allow us to distinguish one group of bacteria from another. Which of these following is a differential stain? And that would be C, the Gram stain. Glycolysis generates ATP by way of, and that would be A, substrate phosphorylation. 39. He developed a post-surgical treatment of wounds. That would be Joseph Lister. And then 40. must be recycled in order for glycolysis to continue in the absence of oxygen electron carriers. To continue in the absence of oxygen, this pathway requires recycling of NAD. And that would be A, glycolysis. And I understand a lot of people miss this, and you put TCA. Even with the recycling of NAD molecules, the TCA cycle does not happen in the absence of oxygen. Only glycolysis happens in the absence of oxygen for aerobic organisms. So the answer here is A. Excuse me.
You said nitrogen and hydrogen are something to be used for.
I'm sorry?
At least nitrogen or hydrogen are something to be used
For an anaerobic microbe. But then in that case, none of these would have been shut down.
Right, okay.
Yeah. 42. This pathway does not produce energy. That's going to be fermentation. The end product of this pathway is lactic acid. Again, fermentation. In this pathway, pyruvate is used as a terminal electron acceptor. Again, fermentation. Pyruvate is the end product in this pathway. That would be glycolysis. 46. And now here, and you'll note on your answer sheets, I had to make some corrections here. Bleach is definitely a disinfectant. Milk and fruit juices are definitely pasteurized. But 48, I went back and looked at your book, 48 is identified as both an antiseptic and a disinfectant, so I accepted both answers on that one. 49 is B, a disinfectant, and again, 50 can be used as both an antiseptic and a disinfectant, so I accepted both answers on that one as well. And all of your tests should already be corrected for that, so you should see that. 51, bacterial, oh, that L shouldn't be there, should it? Bacteria that grow at body temperature, A, mesophiles. Bacteria that can live in the ocean and on skin, B, osmotolerant. Grows best in the presence of oxygen and can grow in its absence, C, facultative anaerobe. Grows well in your refrigerator, D, a cyclotroph. 55 uses fermentation in the absence of oxygen, again, a facultative anaerobe, so that would be C. 56, term that describes the transfer of a plasmid by way of a sex phyllis, conjugation. 57, phrase that describes a disrupted gene when a transboson jumps into it, insertional inactivation. The D value of a chemical reduces the population of bacterial cells by what percentage? 90%. A term that describes an organism that needs many growth factors, fastidious. And what is sweeter than a woman's kiss because you need it for glycolysis? Glucose. And you had to say glucose. Question 61. State the main concept and list the evidence supporting the endosymbiosis theory. So you should have told me that in some form or another that mitochondria and chloroplasts are believed to have one time been free-living organisms that took up residency in a eukaryotic cell and established an interdependency. If you didn't say that much, I gave a lot of leeway here as long as you established that mitochondria and chloroplasts were once free-living organisms. Then you needed to tell me list the evidence. Many of you just said two membranes. Well, two membranes isn't really enough. I think I accepted it for the most part. But the nucleus also has two membranes around it. You should have said two membranes, the inner membrane more similar to prokaryotes. That would have been a complete answer there. DNA, mitochondria have their DNA, their own DNA, and drawing a blank here. Ribosomes, thank you, ribosomes. And then you should mention that the ribosomes were 70S for mitochondria and 80S for eukaryotic cells. And then the last thing that you should have said was that we have identified 4 genes that rickettsia the obligate intercellular microbe has that it shares with mitochondria. And so that really was the nail in the coffin. And so I was looking for rickettsia to get the full 5 points. If you didn't mention rickettsia, you probably didn't get the full 5 points. 62. Diagram and completely label the bacterial growth curve as seen in a laboratory setting. Okay, so your curve should include on the axis, number of cells. If you didn't put that, and time. I mean, if you didn't put that, you would have probably lost points. And then your graph or curve should have looked something like this. I talked to you about making sure that your curve actually represented what's going on in the laboratory. If you started down here and did something like that, we never started with 0 bacteria. You can't transfer 0 bacteria and establish a colony. You always have to start with some bacteria. So you would have missed points for doing something like that as well. And then you had to identify the stages: lag, exponential growth, stationary death, prolonged death. And then I wanted you to tell me what was happening to the growth and why in each of the phases, or what was going on. So lag, you would have said no growth or no increase in cells, and you would have said tooling up or making proteins for growth or metabolizing or something like that. For the exponential growth, you would have said microbe growing at its fastest. You can't say exponential phase, microbes growing exponentially. Can't use the same term to describe the term. So you got to say growing fastest or doubling time at its fastest, something like that. And then you could have also said best time for testing for antibiotics, best time for testing for gram character. Stationary phase. You were going to say that the growth rate is equal or stable or stationary, or I couldn't say stationary, but you could say birth rate equals growth rate. Most of you said that, but you also got to say why. In this case, wastes are increasing and nutrients are declining. And so that's why you see the growth rate slowing down there. The death phase. Death now, or the cell count, is declining at a constant rate. You might even say logarithmically, but not at the same logarithmic rate that you would see over here with growth. And why? Because nutrients have been depleted, waste accumulation is high. Finally, prolonged death is a slow decline in the population until extinction. At some point, they will stop growing. And you could mention that the cells were feeding off of dead cells, so you had a survival of the fittest sort of situation developed here, where the stronger cells were able to survive. So that would have been a perfect answer if you had included all of those. I only had a couple of perfect answers. I didn't give many 10s on that. List 3 activities in which bacteria use proton motive force. Surprised that I didn't get as many. I thought this was a slam dunk question, but apparently it wasn't. So it means I need to emphasize it more next time. But 3 things that the bacteria can get from the proton motive force. Well, they can generate ATP, and that's the main thing that we talked about. But if you remember, they can also use that proton gradient to move molecules across the membrane. Okay? So they can use those, open up those protein channels, and as the hydrogen ions flow into the cell, they can bring molecules with them. And then finally, twirling the flagella. We can use this proton motive gradient, if you will, proton motive force, to turn the the flagella. I'm almost afraid I'm coming down with something. Last question. And nobody got full points for this. This is sort of a test question. Up to question 63, there's been 105 points. So question 64 was a total freebie. Doesn't affect your grade at all. If anything, it gives you a chance to earn a few extra points. Considering the topic of mutation and their consequences, what is the significance of Richard Linsky's work with E. coli bacteria? What I was getting at here was that you would have said something like, mutations can lead to new gene products, new proteins, new enzymes, things like that. selection pressures, environmental pressures can select for proteins that give the microbe an advantage. And this advantage then can allow the microbe to outcompete the other microbes in the population. It's a classic example, underscores completely the evolutionary theory. I mean, it's really the evolutionary theory in a nicely wrapped package. You can see all of the details and context of the evolutionary theory in this one study. That's kind of what I was going for. Review your points, make sure I totaled them up correctly, make sure that your tests are answered or graded correctly, and then
Is it really just the 3 points I mentioned mitochondria like closer